Okay, we are on to the adaptive defenses. I think I'm going to try this in one screencast, but it's going to be a long one, so please remember to take breaks along the way. So just to review, adaptive defenses are specific and have memory. This will make more sense when we get into it. And then it's divided into two arms or branches: your humoral immunity, which is antibodies. Now the antibodies come from cells, but they float around in the bloodstream, which used to be called a humor, so it is humoral immunity. And then your cell-mediated immunity is your T cells. So there are two different types of lymphocytes involved in your specific defenses: B cells and T cells, or B lymphocytes and T lymphocytes. B cells are called B cells because they are born and mature in the bone marrow, and the T cells mature in the thymus. Now I have, there we go, trying to get this to advance, there we go, changed some of the slides, but I have reposted what I changed. So if what you have doesn't match what I'm doing, you can download new ones. Because what I'm working with right now for PowerPoints is what is up on Canvas as we speak. So this, what am I trying to say? The maturation process involves developing what is called immunocompetence. So all of your immune cells need to be able to ignore proteins that are supposed to be in the body. So they are not supposed to treat your own cellular proteins as antigens. At the same time, they need to be able to properly interact with other cells or other proteins in your immune system so that they can communicate with each other. If they fail to do either, they are instructed to undergo apoptosis or programmed cell death, and they carefully take themselves apart either in the bone marrow or the thymus so they don't just break apart, which would cause inflammation, because anytime a cell spills its guts, you get inflammation. Antigen, I don't know where to put this slide. We've talked about it before. It is anything that can mobilize your adaptive defenses, whether it's activating a T cell or activating a B cell. If your immune system responds to it, it is an antigen. So now we get right into what is humoral immunity, or how do your antibodies work. The first thing you want to know about antibodies is that there are two general regions to the protein. Right? So this whole thing, which contains multiple peptides, is one big protein, and this region is called the variable region because it varies a lot from B cell to B cell, and then the end of it is the antigen binding region. And so there's two more things I want to point out here. This is where the specificity comes in, right? So this antigen binding region is specific to the antigen. So one antibody might stick to E. coli, whereas another antibody might only stick to Staphylococcus or Streptococcus. And you might have antibodies to the coronavirus, which are going to be different antibodies than you would have to a cold virus or a flu. So the variable region, there might be, if you have 1,000,000 different lymphocytes in your body, that means 1,000,000 different variable regions until they start reproducing. You do have more than one constant region, but they're not nearly as variable as the variable regions. So you have as many variable regions as there are antigens plus some. Then you have these 5 different constant regions. You do not need to know the constant regions, and you don't have to really be able to explain how they work, but I want you to understand how they work. So the variable region determines where an antibody, the variable region is going to determine what antigen the antibody sticks to. And then your immune system makes several different versions of that same antibody. So let's say you've got an antigen binding site that is an anti-strep antigen binding site. And so you have B cells that are making this anti-strep antibody. They can make the anti-strep antibody in these 5 different flavors or 5 different classes. And if you look here, these antibodies are all connected to each other through their constant regions, and they form this pentagram or pentamer shape. You will see later this makes them really good at cross-linking cell surface antigens. So this antibody right here is the antibody that agglutinates. This is what the agglutinins in your bloodstream look like. And then other antibodies, like this guy right here, slightly different structure. Other thing to point out about it is that for whatever reason, this is the type of antibody that ends up in your saliva. So sometimes the constant region, it determines what the function of the antibody is going to be or how it's going to interact with the antigen. And sometimes it's just different classes of antibodies or different constant regions are used in different parts of the body, like the bloodstream versus the saliva. So then we move into what are the antibody functions. This used to be an essay question, but it is not. So if you find this confusing, you can relax a little bit. So the first thing you want to understand is that for each one of these different functions, it all simply starts with the antibody sticking to the antigen. And then depending on the constant region of the antibody and or the nature of the antigen, right? Is it big? Is it small? That's where the different functions get their name. But they're all very similar because they all have to do with the antibody sticking to the surface of a pathogen, either a virus or a bacteria. And in many cases, they also all, no, not all, they also tend to enhance phagocytosis. So first up, neutralization and opsonization. So an antibody can stick to any protein on the surface of the cell, or of the pathogen. But if it sticks to an important protein on the surface of the pathogen that prevents it from doing its job, we call that neutralization. So here is our nemesis right now, the coronavirus, and we said before that it's these gray spike glycoproteins that attach to the angiotensin converting enzyme 2 receptor on the surface of your cells, and that is how they get inside of your cells. If antibodies were to stick to the spike glycoprotein, they would gum it up. It would be like sticking gum in somebody's car door. Well, nobody has keys on their car doors anymore, but you understand what I'm saying. It would be like putting gum over a lock and then you couldn't put a key in it. So this simply neutralizes or covers up, renders unusable the protein on the surface of the antigen that makes it pathogenic, that gives it its infectious ability. Antibodies can also just bind to any old protein on the surface of a bacterium, and if they have the right constant region, that is going to be yummy for macrophages. So just like macrophages like to grab onto and phagocytose anything that's coated in complement, they will also grab onto and phagocytose anything that's covered with an antibody with a macrophage specific constant region. So then we get into agglutination. Again, this is an example of a constant region or class-dependent action of an antibody. Imagine then you have lots of free-floating coronavirus particles coursing through your bloodstream. What your IgM, I think we said this was, does is it's really good at cross-linking them because they're big and they have, there's 2 on each antibody, so 10 antigen binding sites. 1 antibody can stick 2 viruses together, and then if you have more than 1 antibody on a single virus, it can link all of these 3 together. So you get great big cross-linked clumps of cell-bound antigens, whether you're talking about a bad blood transfusion, a clump of viruses, or a clump of bacteria. They're all clumped together, right? This makes them less able to spread throughout the body, to spread the infection, and it also means that when macrophages come around, they're going to find clumps of pathogens instead of having to hunt down individual pathogens. So it makes phagocytosis much more efficient. Then you have precipitation, and this is simply the same thing we've seen before, but because the antigen is different, we call it something different. So if antibodies stick to soluble antigens, so you want to think small things like here we have snake venom, but it could also be Botox or botulinum toxin. So it's a small chemical that can float around in the bloodstream and wreak havoc. Then what your antibodies do is bind to the small chemical and cross-link it and cause it to clump and fall out of solution or precipitate. So again, two important things have happened here. One, the toxin is bound to an antibody and not to some other part of your body that it might be disrupting. Botox, if I remember correctly, is going to bind to acetylcholine receptors. This is why it causes muscle paralysis, right? This is the stuff that people inject in their face so they can't raise their forehead and get forehead wrinkles. So their skin looks smooth, but they lose facial expression and they're just weird. I apologize for causing anybody weird if I have a student that is doing Botox. But when you see somebody on TV that cannot lift their eyebrows, it is a little unsettling. So at any rate, that's precipitation. It's very much like neutralization, only instead of neutralizing something on the surface of a virus, you're neutralizing a soluble agent and then also causing it to clump. And again, this makes it easier to clean up. So macrophages can come along and clean up and digest large piles of soluble antigens. Then we have complement fixation and activation, which we've heard of already, right? So antibodies can stick to antigens on the surface of the bacteria. Complement comes along, sticks to the antibody. That sets off that big chain reaction, and then the complement is going to opsonize the bacteria, cause inflammation, and poke great big holes in the bacterial wall, ultimately killing it. So now just back to the classes. Again, you don't have to know them. I just want you to understand, right? So this class would be very good at agglutination. This class right here says opsonization. So this, what am I looking at? This constant region right here would be very good at attracting macrophages. And then this constant region right here gets stuck in the cell membrane of B cells where it acts as a B cell receptor. So this receptor right here, you want to think of as being the same as, say, a hormone receptor. I don't know if they work the same way, but they basically do the same thing, where something sticks to the receptor on the outside, and then inside the receptor is hooked up to lots of cellular machinery so that the cell can respond to molecules, proteins, antigens in its environment. So here you have a B cell with this class of antibody stuck in the membrane acting like a receptor that will let this B cell know when it is bumped into an antigen that its antibody recognizes. So now we have to talk about, where do all these antibodies come from? How do we generate when they are naive, as they are here? I don't know if we've covered that topic yet, naive. Naive means immunocompetent, but has not yet bumped into the antigen that it is meant to fight. So if you have never had chickenpox, you have B cells that make antibodies to the chickenpox, but they're called naive because they've never been activated. So you've got, according to sources, about 1 billion different lymphocytes. They each make a unique different antibody. Antibodies are proteins. The instructions for making proteins you can find in genes. However, you don't have room in your cells for 1 billion different receptor genes. So what your B cells do is actually edit and shuffle around the cellular genome. So somatic just means regular body cells. But they edit their DNA, which should just be blowing your mind right now. Like there should be gray matter splattered on the wall behind you because your head just blew up because I told you one of your cells mucks with its DNA. If you're a bio major like I was, you are told from day one that DNA is like the holy grail of genetic information and you just don't mess with it. Every cell in your body has all of the same genes. This is why you can start as a unicellular fertilized egg, and then that egg can divide into different cells, and all the cells can differentiate, and all of the genes that you need to make your neurons and your skin cells and your muscle cells, all of that information is found in the nucleus of that original egg and passed on to every cell in your body, so that any cell in your body can make any protein in your body if given the right developmental cues. So it's just mind-blowing that you have this population of cells that messes with their DNA, right? This is why cloning is possible, why you can take a nucleus from an adult, pull it out, pop that nucleus into an unfertilized egg, and then trick it into thinking it's been fertilized. This is how cloning works. Then that egg, which contains the nucleus or the DNA from a fully grown donor, will develop into a genetic copy or a clone of that donor. Just amazing stuff. So, now that we understand that, and there's kind of two interesting things to take away from it. One is this mind-blowing DNA modification thing, and the other is that just to remember that the raw material that your immune system has to work with in terms of what you're going to be able to fight off and what you're not going to be able to fight off is genetic. So there's a genetic component to how robust your immune system is, or what its potential is. So now we have to talk about how do B cells become activated. And so we want to remember that you have one unique B cell. If it is naive and has never been exposed to the pathogen, then its B cell receptor, its antibody, is different than every other antibody in the bloodstream. So the first time you are exposed to an antigen, this is called antigen challenge, the first thing your immune system has to do is identify among all of its unique antibodies which ones recognize proteins that are part of that pathogen. So you have to find all of the B cells that make antibodies that can stick to different parts of your staphylococcus or your streptococcus or your coronavirus. So step one, when you are challenged with an antigen, is clonal selection. This is identifying the B cells that make the proper antibody. So here we have 3 different naive B cells. Their antibodies are all different color because they're all different. These are all genetically different from one another, right? Largely the same, but definitely genetically distinct. And this one right here happens to have the antibody that can bind to this particular antigen. Now this cell has been made aware of the fact that its antibody can stick to that antigen, and your body now needs a lot of that antibody as soon as possible. So the first thing then that happens following clonal selection is proliferation, right? So this genetically unique B cell is going to make now many genetically identical copies of itself, so that you have thousands of cells now that all make the same antibody. And because they're all genetically identical, we call them clones. It is like the stormtroopers in the original trilogies before other people were stormtroopers. They were all clones of, if anybody remembers, it's Jango Fett. Sorry, a little Star Wars geeky outiness there, but I'm over it. So now we have this clone, then those genetically identical clones are going to become functionally different. They are going to differentiate. Most of them differentiate into what are called plasma cells, and the plasma cells here you can see secrete the antibody. So this is now a different class of antibody. It might be IgA, it might be IgG or IgM, it doesn't matter. And each plasma cell can decide which constant region it wants to stick onto this antibody. So all of these antibodies are going to recognize the same antigen, but they might have different constant regions so they can do different things, or because they're made in different parts of the body, like the saliva versus the bloodstream. So a lot of your clone develops into plasma cells. Plasma cells go off and secrete antibody. Some members of the clone stay behind and become what are called memory cells. So they look like this original naive cell, only now you probably have thousands of them. I do not know how many memory cells you form, but it is a lot more than just one. So that the next time you get infected, the next time you encounter the same antigen, instead of having just that one B cell that makes an antibody you need, you have a couple of thousand, and it's just much quicker to ramp up production if you're starting at 1,000 versus 1. So this is where the immunological memory comes from, is the memory cells. So then what we're going to do is layer sort of what we know about, or I don't know, layer. We're going to add another layer to this, which is your experience of being sick, keeping in mind all this that we know here. Because basically what we're going to be talking about is your primary immune response versus your secondary immune response, and that's what we see here. This is primary immune response, this is secondary immune response, but we're just going to flesh it out and make it sound like what your experience actually is. And these are open-ended questions. Be able to explain the primary immune response and the secondary immune response, and it's just what it says here. So primary immune response, first exposure. So you have a lag period, right? This flat line here is the time it takes to go through clonal selection and proliferation. That takes a few days. So while your immune system is sorting out which B cells it needs, you are getting sick. So this flat line part here is when you are symptomatic and not feeling well, and then after you go through clonal selection, proliferation, and differentiation, you get this immune response that is able to overwhelm the virus, and then you feel better. And then maybe, right, 5-6 years later, maybe a month later, who knows, you get infected again. Or you get exposed again, but now you're here. You have a whole ton of memory cells, and they are able to make a lot more antibody in much shorter time. So you are able to clear the virus from your system before you're symptomatic and before you can pass the virus on to other people. So during your primary immune response, not only are you symptomatic, but you're contagious. And during your secondary immune response, you are not symptomatic and you're not contagious. This, by the way, the nature of the primary immune response to the coronavirus is one of the reasons why we're in such a tough spot, because you are contagious before you're symptomatic. So simply quarantining those people that don't feel good has not been sufficient, because you can transmit it before you're symptomatic. And this is one of the reasons why we've had to go through all of the trouble of having these lockdowns. That, and we didn't have an effective test early enough, and we still don't have enough copies of that test to let people roam around freely, and then test as many people randomly as we can, figure out where the virus is popping up, and then just quarantine and do enhanced social distancing just in the towns or just in the counties that need it. In a perfect world, if we had had a ton more tests early on, we probably wouldn't have had to do nationwide and statewide lockdowns. We could have been much more selective. But we were just not fortunate enough to have a test quick enough. I will move on. Let's see. So yeah, and then this just, again, overlays what we went over here with the immune responses. So primary immune response, all of this is happening. This takes time, so you feel sick. Secondary immune response, you're starting with a whole lot of memory cells, so you produce a whole lot of antibody in a short period of time and you don't feel sick. Next up, acquired humoral immunity. This we will go through as quickly as we can. What did I do here? Oh, I put everything on the same slide, but I forgot to delete a slide. So acquired immunity can be active or passive. Active, as it says here, the B cells encounter the antigen, so the B cells are actually making your own antibodies. Passive means you're not making your own antibodies because your B cells were not actually exposed to the antigen. Then both active and passive come in natural and artificially acquired flavors. So naturally acquired immunity means that your exposure was a natural source. Artificially acquired immunity means your exposure was an artificial source. And when we put it all together, it looks like this, right? So naturally acquired active immunity means you're making your own antibodies because you were exposed to the pathogen in a natural way. This is getting sick. This is the first time you have chicken pox, or the first time you get the coronavirus. And yeah, I won't go into detail, but we may or may not be able to be reinfected by the coronavirus. I don't know yet. So anyways, that's naturally acquired active immunity. If you are artificially exposed to a virus and therefore making your own antibodies to it, so artificially acquired active immunity, that is what vaccines are. So they take, there's two major different kinds of vaccines. One is they take the fully pathogenic, dangerous virus and chemically break it apart so that it is no longer intact and then no longer functional, and then they inject you with those parts, and your immune system only needs to be able to recognize the parts. It doesn't need to see the virus as a whole. It just finds antibodies that recognizes the different parts, and now you can fight it. Another way to do it is to take a more virulent, aggressive strain of the virus, maybe edit the DNA a little bit. You can take some bits of DNA out and make what is called an attenuated strain, or a strain that does not make you sick, and inject that into the person. And if the outside of that attenuated strain, that's what your immune system is going to be primarily interacting with, if the outside of that attenuated virus is the same as the outside of a more aggressive virus, then by being exposed to the attenuated virus, your immune system can also fight the more aggressive, naturally occurring version of the virus. So the natural way to acquire passive immunities is to get them from your mother, either through breast milk or across the placenta. The artificial way to passively get some antibodies into your bloodstream is with a syringe. So it's usually a gamma globulin injection. This is what antivenom is. You get antibodies to the snake venom injected into your bloodstream, and the antibodies precipitate and neutralize the snake venom. All right, now we are on to your cell-mediated immune response. So we're talking about your T lymphocytes two main types of T cells: cytotoxic T cells, which we are going to see have this CD8 protein, and helper T cells, which have this CD4 protein. That will come up again, but I think the more times we mention that, the better. So before we talk about how those T cells are activated, it makes more sense if we know what they do. So the first thing you want to know is, what does a cytotoxic T cell do? What cytotoxic T cells do is scan cells in your body for signs of infection. So they are primarily going after virally infected cells. They can sometimes go after cancer cells, but I don't know how good they are at that, and they can also go after foreign cells. And then they cause the cells that they target to undergo apoptosis, or programmed cell death, so that a virally infected cell will take itself apart before it starts releasing more viral particles, and all of its little parts will be cleaned up by macrophages, and that virus is going to be digested right along with the cell that it had infected and will no longer be dangerous. Then you have helper T cells. What they do is help activate cytotoxic T cells, other lymphocytes, and B cells. It says helper, so it sounds like they're not super duper important, but it is your helper T cells, or your T4 cells, that are the target of HIV. Many of you are probably too young to remember HIV and what it was like when somebody developed actual AIDS, the full-blown syndrome where it's acquired immunodeficiency syndrome. But this is what happens to you when you don't have enough T cells. Your immune system does not work well at all, and it'll eventually kill you. We now have relatively effective treatments for HIV, so it's not nearly as scary as it was. But for somebody that grew up when it first came on the scene, it was a really sobering and scary time. All right, so getting back to the T cells, there are two important receptors on both your cytotoxic T cells and your helper T cells. They each have a receptor which looks like this over here, and if you look just like the antibody, it has a variable region and a constant region. You can think of these as being exactly the same as an antibody, only they always stay stuck in the membrane like a receptor, but they are going to be as variable as antibodies are. That variability is generated by DNA recombination, or editing the DNA. So your B cells, your B lymphocytes do the same thing, your T lymphocytes do the same thing as your B lymphocytes and edit their DNA to generate this incredible diversity of T cell receptors. So the T cell receptor is just like an antibody. It is going to scan or recognize antigens. What the CD protein does is match up different T cells with different types of antigens, or different what are called antigen presenting cells. Let's see. So yeah, just to review, CD4 helper T cells, or I should say the CD4 protein, the cell differentiation glycoprotein 4, is on your helper T cells. Cell differentiation glycoprotein 8 is on your cytotoxic T cells. And the way I remember it is that if you have infection, you want to call for help. So call for help, helper T cells, CD4. And cytotoxic T cells annihilate infected cells. So CD8, annihilate, cytotoxic. There you go. Everybody's going to remember that 100% on the exam. Quick break. And now we talk about T cell activation. It is a lot like B cell activation. It starts with antigen challenge and it ends with proliferation and differentiation. There are two major differences. One is the nature of how the T cells first encounter the antigen, and then the other is this little extra step that takes place called co-stimulation. We'll talk about that when we get to it. So when it comes to antigen challenge, what you need to know is that T cells do not just bump into antigens that are freely floating in the bloodstream the way the B cells do. They have to have another cell in the body present the antigen to them. Any cell in the body can be an antigen presenting cell. We divide them up kind of into these two groups: macrophages, which we want to remember are not necessarily infected, and then infected body cells, which definitely have to die. And the CD protein is how the cytotoxic T cells differentiate between antigens that are being presented by a macrophage and antigens that are being presented by cells that are not macrophages and therefore might be infected. So here you just have, how should I do it? Oh, I'm sorry. It's complicated. Sometimes I forget what I've said and what I haven't said. The mechanism by which antigen presenting cells present the antigen is called major histocompatibility complex, or MHC. So the MHC is a protein that is stuck on the surface of the cell. And what these antigen presenting cells do is take pieces of potential antigens that they want to have scanned, stick them onto the MHC, and then have a T cell scan the antigen. So when an antigen presenting cell meets up with a T cell, two things happen. One is that the CD protein is going to bind to the MHC. So right here, that maroon and that green protein are locking together, and that's what's docking the two cells in an orientation such that now your T cell receptor can scan the antigen and determine if it's foreign or if it's supposed to be there. If it's supposed to be there and the T cell is competent, the T cell will just walk away and not do anything. If that's not supposed to be in the body, that little yellow circle, then depending on what kind of T cell this T cell is going to go about fighting that infection. So now we need to understand the two different types of MHC. There's class 2 and class 1 MHC. Class 2 MHC is for fragments of extracellular proteins. So this means it has been engulfed and digested by the phagocyte or by the macrophage. So here you have a virus, here you have a macrophage. The macrophage is going to engulf the virus. Here it has digested the virus. So this macrophage is not infected. You do not want this macrophage to be killed by a CD8 cell or a cytotoxic T cell. So these little chewed up bits of virus are taken over to another organelle that has the MHC in it. This is budding off of the Golgi, probably, or maybe the rough ER. I don't know exactly when this takes place, but little pieces of the virus are complexed with the major histocompatibility complex, and then that gets moved to the surface. So now you have this little chunk of virus that has just been killed by the macrophage presented on the MHC2. So this macrophage is basically saying, hey, I just killed and ate this thing. Is it a virus? Because if it's a virus, we have a problem. If it was a dead skin cell, it's not a problem. Then MHC1 is for fragments of proteins that were synthesized inside the cell. So if this is a virus here, this virus is inside the cell, this cell is infected. And what all of your cells do is randomly sample proteins floating around in their cytoplasm, chew up the protein, complex it with MHC1, and then stick that on the surface. So this could be a protein from inside the cell that's supposed to be there, in which case this antigen will be ignored. But if this MHC1 is presenting a piece of virus, that means that virus is on the loose inside that cell. This cell needs to die because it's infected with a virus. Oh, I forgot I left this here. So just to review, right? MHC1 presents the antigen, and make sure that the right T cell is scanning the right MHC, or the right antigen presenting cell, and that allows the T cell to scan the antigen and determine whether or not it is a foreign particle. So put it all together, here you have a macrophage. It is going to be using MHC2 because it is presenting antigens that it has engulfed and destroyed. It is going to present its antigens to helper T cells because the CD4 is going to bind to the MHC2. And then the T cell receptor will scan the antigen. Over here, this cell is infected because the virus is floating around inside its cytoplasm. So those viral particles are going to be presented on MHC1. MHC1 is going to dock with CD8. So this is going to bring the cytotoxic T cell receptor in close proximity to our antigen, and now this cytotoxic T cell is going to kill this infected cell. Well, we're not quite there yet, so I got ahead of myself. That is antigen presentation. So antigens have been presented to our helper T cell and our cytotoxic T cell. The other thing that has to happen at the same time is that some other cell, probably a helper T cell, is going to dump immune chemicals onto our cell that is being presented with an antigen. And does it say? It does not say. These are, oh, no, it does say, I'm sorry, interleukins. Interleukins is a class of chemical that white blood cells, or protein that white blood cells use to communicate with one another. So this cell here gets exposed to a potential pathogen, has interleukins dumped on it. That makes it now fully active. It is going to proliferate, form a clone. Some of the clone is going to go off and fight, like that's what these cells here are doing, and some of the clone is going to remain behind as memory cells. So if you are a helper T cell with your CD4, when you become fully active, what you do is dump those immune chemicals on other cells. So you are part of the co-stimulation process. Here they are calling them cytokines. Cytokines is the broader term for immune signaling chemicals. So histamine, interleukin, I think colony stimulating factors, prostaglandins, these would all be considered cytokines. And it's a term I don't use very much because I don't want to introduce too many terms, but it's there, so I thought I'd describe it. So you fully activate a helper T cell, it goes around releasing cytokines on other cells. You fully activate a cytotoxic T cell, it then causes apoptosis in the cells whose antigens it recognizes. Just remember that the cell doesn't really blow up like that. It folds in on itself and forms like a bunch of little smaller bubbles. I forget, there's a name for it. But it basically takes itself apart very carefully. And then a macrophage will come over and phagocytize all of the little bubbles that this cell has made of itself. So that's the most complicated part probably of the whole class, is understanding B cells and T cells, and especially T cells because of MHC1 and MHC2, and it's complicated. But just keep reviewing that. That is something that I don't think for most of you is going to make sense the first time through. I usually remember to say this in the classroom. Sometimes I forget when I'm just talking to a computer. If you are confused right now, that is normal. You should not be feeling bad about yourself if you find a lot of this to be confusing. This is why you have homework and studying and study partners and the book, because a lot of us, myself included, don't get these complicated stories the first time through. So don't worry if you're confused, just go back and review it until you're not confused. What do we want to know? Yeah, probably won't ask too many questions about this, but it is nice to remember that the non-memory actively fighting T cells, whether they're helper T cells or cytotoxic T cells, do not last forever because they can wreak havoc if they're still going around doing what they do after the infection. Remember, it is your immune system that causes inflammation. So if you have helper T cells churning out more pro-inflammatory chemicals than you need, then you're going to get a lot of post-infection inflammation. It's just going to make you feel bad for a longer period of time. Regulatory T cells, you just want to know, exist, and they help dampen immune responses. And I don't have anything more intelligent to say about that. Hey, now we're revisiting natural killer cells. Remember when we first looked at the natural killer cells, we say they just kill anything that doesn't look right. But now we know what the look is, right? So some of your cells, some of your viruses, and don't ask me how they do this, but one of the things that can happen when a virus infects a cell is that the cell can pull MHC1 from the surface so that your cytotoxic T cells don't have anything to scan. Your natural killer cells come along and they go, hey, no MHC, you gotta die. So they eliminate the cells that are trying to evade detection. They can also, as this says here, target cells that are coated with an antibody. I'm not really sure how that works. And then different surface markers of stress. I do not know what the surface markers would be, but you could imagine that if your intracellular machinery is working overtime churning out virus, you are going to be metabolically stressed. This is the same for cancer cells too. Some of your tumors, if it's a very fast-growing tumor, those cells are dividing faster than cells are supposed to divide. This is causing metabolic stress in the cell. Sometimes that shows on the surface, and that's one way that your immune system can notice and target potentially cancerous cells. I don't know that we have a really firm grasp on how good our immune system is at killing cancer or precancerous cells, because we only know about the problematic cells until they develop into cancer. We don't know how many successes your immune system has because we never notice it. Immune deficiencies, they are just immune deficiencies. I will not ask you to define them. The one thing I will ask you to remember about HIV is that the cell that it targets is your helper T cells. And then all of these here, just read over that short paragraph and make sure you can recognize all of those as autoimmune diseases. Hypersensitivities, these are allergies. I'm not going to ask you much about allergies. I just want you to know the difference between type 1 and type 2 hypersensitivities, or acute or delayed hypersensitivities. Acute hypersensitivities happen quickly. And generally are mediated, or the effects, the symptoms of the allergy are caused by histamine release. So type 1, fast-acting histamines. I'm not going to ask you about shock. Just know that anaphylactic shock is bad for you. This happens when you have immediate hypersensitivities and it's systemic. So basophils and other immune cells throughout your body are releasing lots of histamines. Then all of your blood vessels become vasodilated and permeable. So fluid starts leaking out of your bloodstream. Your blood pressure drops. It goes in the dumper. And so you can go into shock because your brain is not getting enough blood. Plus, it can also cause constriction of your bronchioles, and then you can't breathe. The treatment for that is an EpiPen, which is going to cause vasoconstriction of all of your blood vessels so that they they go back to normal. Let's see, and then delayed hypersensitivities. This you just want to know is your T cells. And presumably it's because they have to proliferate and mount a response, and it takes a little bit longer for T cell action to cause hypersensitivities. But this is when your T cells react to something like poison ivy the same way they might be reacting to a virus or a bacterium. And that's it. See you in the next screencast.